Today's presentation is going to be on defining host pathogen interactions, employing an artificial intelligence workflow. This is Fisch et al. In the lab of Ava Frickel, and that is at the Francis Crick Institute in London, which is a pretty prestigious institute. And this was published in one of the big four journals. So we did our last two coming out of Nature. The other big four are Cell, Science, and eLife. So this is eLife. And so we have high expectations for the quality of work that would be published in a journal of such high regard. Okay. So without further ado, Katherine, go ahead.
All right, just want to start things off. First two papers, really nice. Well, amazing, in fact. I wanted to be a little bit different. So I guess you'll see that as, time goes on and all that fun stuff. So, yep. Can you guys see the slide presentation? Just thumbs up. Yeah, all right, coolio. Yeah. So basically, currently image-based infection biology often uses humans and like human comparison, all that fun stuff. But this is going to use a computer, which is often faster and usually less biased. Is that really going to happen? I don't know, since programmers often implement their own bias when creating something, but that's a whole other can of worms. So basically, this paper goes over the host response to microbe analysis, which I'm going to call Herman for short throughout this entire paper. And basically, it's an open-sourced imaging platform based on machine learning and algorithms with deep learning and all that fun stuff. And it has the ability to learn phenotypes from the data without relying on research-based assumptions. And in this paper, they use the examples of Toxoplasma gondii and Salmonella enterica typhimurium. So yeah, on to the next slide. So the little image on the left, that's Toxoplasma gondii, and it is a single-celled parasite. And the image on the right, that is Salmonella, and it is an anaerobic gram-negative bacteria, usually causes food poisoning and all that fun stuff. So figure 1 demonstrates the actual pipeline that Herman goes through when processing an image. And before it even gives you the results or anything, it has to first receive the image and has to go through user input. This is where the bias may come in, but they try to be as unbiased as possible. And then it goes through a series of image correcting in order to go and make sure that the image is readable by the actual system. And then it goes through the whole entire neural network of determining clarifiers, like how many vacuoles it has, the percent of infected cells, all that fun stuff. And then at stage 2, Herman uses something called the CNN HERMA AlexNet, which was derived from the AlexNet architecture in order to determine these phenotypic characteristics. And then finally, this data is written into a spreadsheet for the researcher to go and have the descriptions of the pathogen and its interaction with host factors in the whole entire population itself and in single-celled areas. So figures 3 and 4, a lot of stuff. Honestly, not too important. I mean, it's important, but basically uses HeLa cells, right? They were stimulated with 100 units of interferon gamma, which is usually the immune system's first line of defense. It warns the other cells around it like, hey, something's wrong. And the left figure, figure 3, uses Toxoplasma gondii, while figure 4 on the right uses Salmonella. And this data would be nice, right? But since they didn't really compare it to anything else besides itself, it's not too useful, right? But they fixed that with figure 5. This, they actually compared it with other, other stuff. This was actually not originally part of the original article. After I went through some research and all that fun stuff, people were like, hey, we really need this in the paper. Can you please put it in? And so the people actually did. And it compares Herman to other platforms, and it uses GRA 16, GRA 24, and TGIST, which is a parasitic protein that's secreted. And the figures themselves, like little graphs with red dots on it, they only tested 3 points, which isn't really the best. And they only tested basically the max, the min, mean values, all that fun stuff. Not really, but yeah. And basically conclusions and possible implications. This is all new. Benefits of machine learning have not been fully realized, but this is growing. This field was like budding in the beginning of like the early 2000s and still hasn't reached its full potential. But basically, what it could do is it could tell a person if they have cancer or not before any of our modern day stuff could reach to that conclusion. And basically, this paper just demonstrates a little stepping stone for what could possibly happen. As for the actual paper itself, I honestly have no idea. And that's it. Yay. I'm sorry.
Wait. So go back to figures 3 and 4, and this is,
Right.
we talk about when generating a hypothesis, the most basic hypothesis for anything is this is different from that. What should have been the this and what should have been the that in this study? What should have been compared in this study?
Anything as simple from the actual Herman platform to human comparison. There could be that option.
That is it. That's it, right?
Yeah.
Our platform with AI is different than or is significantly better than this other platform, right? So what's wrong with figures 3 and 4?
Doesn't do that at
It
all.
doesn't. It has nothing to do with it at all. I think it's worth listen. Journal Club's not. This is directed at everybody else, not Catherine. Journal Club is not about just looking at the stuff that makes sense. It's also looking at the stuff that doesn't make sense to figure out why. So if you look at figures 3 and 4, there's lots of them. What are they trying to tell you, and what does that have to do with the goal of the paper?
How good their AI system is?
That's the goal of the paper, right?
Yeah.
Right. That's the goal of the paper. Do figures 3 and 4 actually do that? And I would love somebody to argue with me that they do, because they don't. Okay. Does someone think that these figures actually show how good their system is versus the status quo?
No.
Nobody wants to argue that, right? Well, what is okay, fine. Let's all remember that Mr. Fisch has Mr. Fisch? Yeah, Mr. Fisch doesn't have his doctorate yet. Has a paper in one of the finest journals in the world, and none of you do. So there must be something to this, right? Let's presume that there is something to this paper that's actually worthwhile. What is it that figures 3 and 4 are trying to show? Or well, what is it that they show?
They show the different parasite loads, infection levels, vacuole position. Shows a lot of different things.
Okay. Measured how? How'd they measure it?
I have no idea.
Catherine?
Excuse me? Sorry.
How do they measure it in 3 and 4?
I think they're trying to refer to like the original.
No, they don't. That would have helped.
Yeah.
No,
That would.
they measured it. They measured it with Herman. The thing that they like. If I walk into a room and say that I'm smarter than a 5th grader, do you just nod your head and go, yeah, you're smarter than a 5th grader if you're a scientist, or do you actually need to also meet a 5th grader and compare the two of us? Right? They did the here Herman. This is how Herman works, and didn't compare it to anything.
So how would they
That's
get away with
inexcusable.
It's inexcusable.
how would they get away with publishing this paper without comparing to a control?
That's where figure 5 comes in.
but it is. I don't know. I said to Catherine earlier today, I was like, if you'd seen my face while reading this paper, I was, I'm reading it, and then I was like, because I did this stuff in my first postdoc. I actually won a grant, $10,000 or something like that, to buy a machine that did this work. It's a typical cell counter, okay? Does things like segmentation, and I had to use a program called Metamorph, and I was doing this on the regular more than 10 years ago. I'm like, so this really isn't that novel, first of all. Secondly, they never compare it to the baseline, to those machines like I had 10 years ago, to show that it's better than that. They never did that. They just showed that it worked. Look, it works. It can identify that. Cool, but I don't know if does it take less time? Is it less money? Like what's so good about it? But I'm reading it. I'm going, this is not a novel thing. This is not, to me, it's not new and interesting. I actually read it twice because I'm like, I must have missed something. Because this isn't an eLife. eLife is run by humans, and they can make mistakes. So like Nature published a paper, Nature being, nature, published a paper. Now I want to say it's probably about 10 years ago, probably around 10 years ago, where a young faculty member at the Riken Institute in Japan published a claim that if you take normal skin cells from a human and you dip them in a certain type of acid, they will immediately turn into pluripotent stem cells. It was a wildly outrageous claim. And when you have outrageous claims, you better have substantial evidence for it. This paper had been rejected outright by Science. The journal Science said, absolutely not. You have not shown this at all, blah, blah, blah. Nature, in their need to publish something they thought was hot, went and published it anyway. Over their reviewers, the editor made the decision to publish it over the reviewers' objections. So usually you get 3 reviewers per paper, 2 main reviewers, and then usually like a third supplementary reviewer. And 2 of the 3 reviewers, I think, on that paper rejected it outright, and the editor accepted it anyway. And then it turned out that when they looked at her data, she had made the whole thing up. Because the pressures of getting tenure were so high, she needed to have a paper in one of the big 4 journals, so she went and did that. Okay. Similarly, and I can never remember his name, but the guy who claimed that autism is caused by vaccines did exactly the same thing. Okay, he published it in one of the most famous medical journals in the world, The Lancet, or the New England Journal of Medicine. It was one of the 2. So and got, it got soundly rejected in the process, but they published it anyway and made a whole generation go by where kids aren't getting vaccinated, and it turns out he made the data up. Okay, I'm not saying that these guys made the data up because this video is going to be posted online. I don't understand. I was reading this and I'm going, why is this in eLife? And if you look at, Catherine, unshare your screen for a moment. I got the paper here. So if you look right here under the abstract, it says editorial note. This article has been through an editorial process in which the authors decide how to respond to the issues raised during peer review, which suggests there were issues in peer review. There always are issues in peer review. Nobody accepts a paper outright. The reviewing editor's assessment is that all the issues have been addressed. Now remember, Catherine said the figure 5 wasn't in the original paper. And that is mind-boggling if you read what was written. So I actually have that. Here it is. This is the decision letter after the peer review. I don't know if you could see this, but thank you for submitting. Summary. Let's see. Major concerns. The reviewers raised some concerns regarding the technical novelty of the analysis pipeline because it's not new. It's not novel, right? In particular, analysis performed in stage 1 is quite standard and follows similar methods described before. Okay, so figure this out. You got everything's connected here. You need to you have to be a detective. This paper was published. Catherine, check on the side of the paper. Was it February 2019?
Yeah, February 12th.
Yeah. So February 2019. This other paper came out in 2018, and we actually looked it up. Here it is. This came out August 2018. Miss Liff, what do you think happened if you're there and want to play along? Nope, she stepped away. Okay.
No, I'm here. Wait. Say that. Wait, what happened to wait.
Look, so their paper, defining this paper was published in 2000 in February 2019. The review says, why didn't this is not novel because of methods described in Toquette et al. 2018. You go to that paper and look when it was published, August 2018. What do you think happened?
Oh, a little copying of someone's work.
They got scooped, right? Like they had this ready to go, and it would have been novel and it would have been cool, but this other paper came out.
Or they honestly omitted figure 5 and had to go through so many peer reviews and add it back in before publication.
Yeah.
That the other paper scooped them.
Yeah. Well, what's weird, it's not really showing on the screen that well, but what's weird about this is this original paper, which is supposedly more novel, was published in PLOS One. I've been a guest editor for PLOS One. I've published two papers in PLOS One. I think it's a good journal. I don't think it's a great journal, but it's a good journal. But it's not eLife. I mean, this journal's way, way better than PLOS One is. So how did this paper that wasn't novel get published in a journal way better than the one that was novel? Right? So then you go on to read, the analysis in stage 2, although well performed, does not represent a major methodological advance in the field of machine learning. Machine learning and AI was the reason, Catherine, right, that you chose it. So she got it was a bait and switch. She got baited with, look, AI, how cool. And then figures 3 and 4 here, figures 3 and 4 had nothing to do with AI. These were biological data. And they were biological data showing things that are well known for 30 years, 40 years maybe. So it's not novel either. So if we go back to here, although the authors present some applications of this analysis pipeline, it would be advisable to provide additional data demonstrating that Herman can detect additional previously established phenotypes, such as those for rope 18 and rope 5, which you're like, what is that? I don't know what that is either. That's not the point. The point is that it can show that these proteins are induced or something like that. And if you go to figure 5, that's exactly what they did. Rope 15, rope 18, right here and here. So this figure was generated in response to reviewers 1 and 3, which means that figure didn't exist before. So all they submitted was this figure, which was a methodological figure, like how to do this thing, this machine learning thing. It's like, we're going to present, like, this is fine. I have no problem with this being figure 1. And I would advise that it be figure 1 in a methodological paper. eLife doesn't publish methodological papers. eLife publishes stuff about new biology, like new findings, things that are novel, things about understanding how life works, not about how to understand, right? Like, not about the method to get there. So this is fine. The weirdness is when all of a sudden they leave all that behind and just show some biological data that has nothing to do with it. Catherine, I said I was going to try to shut my mouth, and I didn't. I failed again. Any thoughts? Smart thoughts? Dumb thoughts? Thoughts in general?
So, like, what was the actual appeal of this paper to eLife?
I actually do have a question, Dr. Char. So, for
let's come back. Let's come back to RC's question at the end, because I think that's kind of how we want to end it, RC. Go ahead, Johan.
So, I have a question for like figure 5. So,
Okay.
Is it like comparing the Herman process, AI process, to like parasitic, like is it, I heard like something about parasites. Is it comparing it to like hold on a sec.
Catherine, this is all you.
All right. Well, Toxoplasma gondii, that's a parasite. So that may be where you heard that parasite keyword.
Yeah.
Yeah.
So what's the purpose of like B in figure 5?
I believe it just shows like comparing the data from GRA 16, GRA 24, and TGIST. That just shows like parasites per cell and then normalized number, all that fun stuff. So I think it's just showing the data from the Herman process through 3 different ways.
Well, I mean, B is a human process.
B is
right?
the human process.
So if you come into a doctor and you say, I've been having these symptoms, okay, you list off all the symptoms, and to the doctor who's gone to medical school and hopefully has some diagnostic knowledge, they'll go, okay, those symptoms sound like toxoplasmosis, so we need to confirm that. So what they do is they take a sample of your blood and then they put it under a microscope after they stain it, and this is what they see. That is a time-consuming process, and it's open to human error, right? So then wouldn't it be great if, finish that sentence, wouldn't it be great if what?
A computer could do it faster and more accurately.
Right. And so you're looking for matching between the eyeball version of it and the computer. There is an inherent problem with the way they did this, which Catherine already mentioned, which is, What's the problem?
Lack of comparative data.
Well, they're at least sort of trying it. It is that is kind of the answer also. That they really didn't do a very good job comparing it anyway. There's something else wrong with it.
Did they not do the comparison?
If you read the bottom of the figure legend for B, overall number of analyzed cells are indicated in the graphs. Data represented as a mean plus or minus SEM with an N of 3 experiments. With an N of 3 experiments.
There was only 3 tests. There weren't several.
They are claiming that their statistical power was so great they didn't need any more samples, a larger sample size. And out of three, they did something 3 times. So, and the funniest thing about this is they have error bars. So if you, well, let me pull it up. If you're looking at this red dot, that's the mean right here. Mean for 2 cells, right? Or this is parasites per cell, right? Cells with 1 parasite, cells with 2 parasites per cell, 3 parasites per cell, 4 parasites per cell, right? But they only have 3 per group. So you're looking at the mean and then the most and the least, and that's it. And look at Miss Liff is laughing very hard. That's really terrible. Like that's not how statistics are done at all. And they let this fly.
So I'm just going to point out, when I did my master's paper, now mine is totally not biologically based, definitely not, well, kind of AI based. I did all satellite remote sensing data. I had over 600 data points, and I was told my data points were light, right? I didn't have enough, according to some of my journal reviewers. And they got away with 3. Oh.
When I did my paper, the Nature Neuroscience paper on seasonality, it was the largest data set ever collected on single cell data, and it was still just barely strong enough statistically, and that was 1,500 cells. 1,500 cells versus 3. And that's, I don't know. So figure 5 was clearly just thrown together at the last minute, right? Because they only collected 3 per, and it wasn't that big a deal. They threw it together, they sent it in, and we're like, are you satisfied? And for some reason, eLife said yes, which makes people like me bitter for a couple of reasons. You know, if you publish in a sexy journal, your career is made. You know, you get grants easier, you get other sexy journal publications easier. I have a paper in eLife that's a pretty good paper. I got it after I had left academics. I got it in my first year of teaching high school. It finally got accepted. Had it gotten accepted 8 months before, I'd be at Harvard right now and not at Morris Catholic, which would have been a mistake. Morris Catholic, much better than Harvard, I hear. Okay. But I didn't get into nature when I sent my paper over there. I didn't get into science when I sent my paper over there. This got into
Fisch, is Mr. Fisch a previously authored paper from eLife? Did you look?
Does he have a different paper? I don't know. Well, wait a minute. So,
because folks, nepotism is strong among any sort of place.
That was kind
so
of RC's
if he's been published before, or if he has colleagues, peers, or a networking in that's been published previously, it's going to be easier for him to be published as well, even with a shoddy paper.
Yeah. And it might not be him. It may be Ava Frickel. I mean, the Crick Institute is a big deal. And speaking to RC's question, like how's it get into eLife? Well, not the editor in chief, but the section editor is an acquaintance of mine, and I directly sent my paper to her so that she could review it, because she's going to win the Nobel Prize in the next decade or so. And, you know, that's There's the nepotism, right? She advised she advised two of my advisors, my college advisor and my postdoctoral advisor. She was on dissertation committees for both of them. And so like I sent it to her. That's just kind of how you play the game. But I think that it's the Francis Crick or Frickle. Let's look that up real quick. Let's see.
Well, I just did a quick search of his name in eLife, and his name comes up 335 times. So, yeah,
Okay.
that's interesting.
Yeah, well, there's. I mean, Daniel Fisch is kind of a very common name. Okay, so Ava Frickle is young. It looks like. So let me look up. And did you look him up specifically in that, Daniel? What is it, Daniel? What? Frisch?
Fisch, I think. Yeah.
CH. No, Daniel Fisch. Maybe you're right. Maybe I was looking. No, he's a young guy. There's no way he has 200 papers. Sorry. He's got papers in some okay journals. eLife's the best one. Embo's not terrible. I don't know what Wellcom is. I've heard of it, but it's like not a well-known. Methods is not a well-known. Plus Pathogens is good. Cell Reports is that's a better one, I'd say. And here, August 2020. Interesting. Interesting. So, yeah, he's a
How
PhD.
much?
So Mr. Fisch. He has not finished his doctorate yet.
How much, like, I just like in general, like, how many as papers are there, like, regarding like things with AI? Like, is that like a big thing at the moment, or is that kind of?
Go ahead, Catherine.
No, it's not.
Yeah.
Yeah, I tried finding a paper that was more AI oriented because that's what I want to get into. This was the best article I could find.
So this is a bait and switch, right? I mean, like, it's like, we know that this is really hot right now. Let's say that we did this, sort of do it, and then maybe we'll get in a high profile paper. That's a really
Part of it could be the naming of it, of using AI as opposed to like computer programming or programming statistical analysis or whatever, right?
Yeah.
Just the naming of what they're using, that could be a big reason as to why you're not finding papers or you're not finding the material that you want to find.
Could that also be one of the reasons why I was picked up by, like, eLife?
Yes. Yes. Yeah. There was the most egregious example of this I'd ever seen was a colleague of mine in Manchester, so right near here, published a paper in Science that said that a very common chemical that you've all heard of called cyclic AMP, CAMP, C-A-M-P, right? I've mentioned that in every one of your like courses at some point. Cyclic AMP does a thing that was obvious, and that was the title of it. It was like cyclic AMP involved in circadian rhythms. No kidding, really? And it got into Science, and then it turned out none of the figures were about cyclic AMP, and we were just like, well, how did my advisor? I really in the senior author on that paper we really like. He's a good guy, and you play the system and he won that particular one. He got his paper into Science, and it really wasn't that interesting. But somebody like my advisor, Doug McMahon, has never had a paper in Science, Nature, or Cell, and has really extraordinarily good work, but never gets a nod, never gets anything in. Closest he ever came was my paper, which got 2 out of 3 reviewers loved it, and then one of them was like, but it's a paper about and named something that had nothing to do with it. So that happens a lot. You name it just right. And I think that's inappropriate. So when you write lab reports in my classes, I say it needs to be in a sentence where you state what the result is, because I want you to be honest. I don't want you to sell yourself. Like that's, I mean, there has to be some selling yourself on the networking side. If we weren't in COVID this year, we would have traveled to Washington, D.C. In November for the Society for Neuroscience. I would have taken you all to the conference. And there you get to rub elbows with all those big scientists and get to meet people. And it's a lot of fun. And you're free. I let you go, right? But so those of you who are freshmen might get to do that 3 or 4 years from now. The many of you who are freshmen. The 4 of you who are freshmen. Katherine, you also went and searched for the website, right?
Yes, if you go hrman.org, herman.org, you'll be able to find it. Yeah, it's not the best looking website, but you know, it's there.
Yeah, this is it. I mean, this is a really very bare bones website about Herman. So if we go back to Katherine, share your screen again, go back to your figure 3 and 4. We're not normally going to do this where she can just say, skip it. But this really doesn't have anything to do with the study. Journal Club is about actually stepping through each one of these little figure sublets to show what it's supposed to show. But it doesn't have anything to do with this, with the reason she was interested in the study. And they're not novel results. So you should look at them and see why. Like if you don't recognize a word, take 10 minutes, look at it. If you don't recognize a word, go Google it and see what it is. You'll find that a lot of the results here are old. They're nothing really that new. Not novel findings. So I'm not sure why that would be important. It's almost like they tried to distract you from the lack of comparison data between human eyeball looking through a microscope and their high throughput machine. Right? Because ultimately, Katherine, what was the point of this paper? What was supposed to be the point of the paper?
Look at this nifty thing and all the cool stuff it can do. It's the future. The future is now.
Yeah. So did they do that?
No.
Okay, go to your implications and conclusion slide. So you seem to have separated this between, click again. Yeah, your expected implications. So you'd actually say like 1 through 4 you actually wrote before essentially reading the paper, right?
Yeah, you could say that.
Based on the abstract and based on your interests. That should have been the implications. Were any of those well-founded?
No.
Not really. It's a different type of paper from the last 2 weeks, or the last 2 journal clubs, right? And you know, I kept saying to Katherine, like, that's not bad. That's not bad. She didn't do a bad job. Okay, this is a bait and switch, and this happens. You know, quite often. I'd say more often than not, the paper's bad. It's usually bad, and every once in a while you get something extraordinary. And actually, we just lucked out and had 2 kind of extraordinary papers the first 2 journal clubs of the year. 3rd journal club of the year, not an extraordinary paper, but a good exposure for you guys to see that everything's not always perfect.
Keep in mind, a bad paper isn't always just a bad paper. You can always use a bad paper when writing a paper as a what not to do.
Mm-hmm. Mm-hmm.
And that can be one of the most useful things. And I can't tell you how many times I've read papers where there has been literally 1 useful sentence out of the entire thing. And I've
right. Jordan, Yeah, Jordan, are you seeing that in your reading? Because Jordan's doing a capstone right now, reviewing papers.
Yeah, they're kind of a hit or miss. There's some you get like a lot of good points from, and then some it's really nothing, and you have to keep looking.
I think that it's also really, it could be really useful because if they missed an opportunity, you have a chance then to do the thing that you wanted to have read in the paper, right? So like in January of 2007, I reviewed a paper for current biology that really struck me. Like it was I thought it was a really good paper, but left a lot on the table. Really good paper. So I approved it. I mean, current biology is a great journal. The impact factor is like 15. Great journal. And I approved it for, you know, I reviewed it, or I approved it. I thought that it was a really good journal article and that it would come out. But I'm like, my system that I had at Vanderbilt was better at finding the answer to that. So I immediately took some animals, ran them through their paces, and that ended up being my Nature Neuroscience paper. So I got a really, really good paper out of it because I paid attention to the things that were missing in a good article. But I mean, especially in a bad article. In a bad article, it leaves you a lot of empty space to work. Because in your field, let's say that you guys go to college and you join a biology lab or whatever, right? You know, a marine biology lab. You're not going to be reading these type of studies necessarily. You're going to be reading marine biology based stuff. If you join a neuro lab, you're going to be reading neuro based stuff. In this, we're trying to, you know, run the gamut of all the possible sciencey things. Like eventually, somebody might do a chemistry paper. Somebody might do a, you know, a physics paper, astrophysics or something. Okay. Do you have anything you want to add, Catherine?
Well, just reading the paper in general, like I found that they were trying to fluff it up a lot, and I guess that was kind of a sign that, hey, maybe they don't really have a lot to say, like in the paper itself. So.
Yeah. You can stop sharing your screen. I hope you guys found that. I thought that was kind of fun. Hopefully you found it worthwhile and not a waste of your time. All right, good job, Catherine. We got to come up with a new system for that. We should all just like raise hand or something like that. You know, in the deaf community, this is an applause. Yeah. So, okay. Thank you very much. Go enjoy the rest of your night.
Have a good one.
See you guys.
Thank you.
Later.
Have a good night.
Yeah.
Thank you.
Thank you.
Thank you, Dr. Char.